So my name is Lana Haganovich, and I'm going to be presenting a study that looks at the influence of the diamondback moth's gut microbiome on its herbivory. Herbivory being defined as the capacity of the moth to consume plant matter and function as an herbivore. Here is the title of the study: Metagenomic sequencing of diamondback moth gut microbiome unveils key holobiont adaptations for herbivory. So to start us off, I'm going to give a little bit of background information. So the diamondback moth, the scientific name being Plutella xylostella, is one of the world's most destructive pests, and it's very resistant to insecticides. It feeds on cruciferous vegetables like cabbage, broccoli, cauliflower, and it's a globally known massive pest in the world. It even co-evolved along with the cruciferous vegetables, which shows how dependent it is on these vegetables, which, of course, people rely on because of our agricultural needs. So these moths spend their entire lives around the host plant. They lay eggs on it, and the problem being that they're resistant to most insecticides. So the authors of this study actually did a previous study that showed that the moth has the ability to detoxify plant defense compounds, which help it work as a pest and keep feeding on those vegetables, because plants, of course, have certain chemical compounds built into them that serve as defenses. But certain insects are able to bypass these defense mechanisms. And this study is specifically looking at the diamondback moth and how it's able to do this. Certain gut bacteria play a really important role in the breakdown of plant cell walls, the detoxification of plant phenolics. Phenolics are certain defensive compounds that plants have, and the synthesis of amino acids, which work to nourish the moth itself. But this study concludes that the previous study that the authors did was actually insufficient, and this study will actually go to create a more complete taxonomic and functional profile of the gut microbiota, meaning it's going to break them down into taxonomical units, classify the microbes into those taxonomic classifications, and look at the functions of those microbes and the enzymes that they have, and the mechanism by which they work to promote the herbivory of the moth. And the study is specifically going to be using metagenomic sequencing. So some enzymes support amino acid synthesis, others support digestion and detoxification functions. But this is really important because if the study finds certain mechanisms that work towards making the moth herbivorous, that can actually be a target for future research to try to figure out how to, target these mechanisms and make them less able to proceed, and therefore stop the herbivorous attack of the moths on crops and help farmers and people who need the agriculture. So past studies have proven that other insects have these capabilities, like termite gut bacteria helps them break down cellulose, which is in plant walls, and obviously breaking it down is bad for the plant. Gut bacteria in honeybees helps them biosynthesize vitamin B12, which helps the honeybees themselves, and degrade pectin. Pectin being a substance in plants, so degrading the pectin serves as eating the plants. And bean bugs actually have a symbiont called Burkholderia, which helps the bean bug degrade an insecticide known as phennylethrone. And this study is specifically going to be looking at holobionts, and the holobiont is known as a term that holobiont is a term that defines the unit of the individual animal and all of the microbes in and around that animal. So it treats it as a single unit, and that's mostly what the study is going to be looking at in these moths and the gut microbiota. So because of what we know about the diamondback moth, the fact that it's resistant to so many insecticides, the fact that it's such an awesome pest, researchers decided that they should look at the gut microbiome and the role that the gut microbiome plays in the herbivorous mechanism. Because, like I said, this could provide like almost a chink in the armor for that microbe, for the moth, which for future research to attack it and then try to suppress these capabilities. The thing is, many previous studies have looked at bacteria in the gut, but they usually used culturing, which is when you multiply microbial organisms by letting them reproduce, like in a medium, or denaturing gradient gel electrophoresis, DGGE, which is a technique that separates a mix of DNA fragments according to their melting point. But the authors of the study determined that the disadvantage of these techniques is they only partially demonstrate the diversity in the gut microbiome, but they want to look at it holistically. They want to get a full, complete taxonomic profile of the gut microbiome in these moths. So, therefore, instead of using the methods of their previous study, which is where they sequenced V6 region of 16S rRNA, they actually ended up using metagenomics. And metagenomics is particularly special because it looks at all of the nucleotide sequences in a given community of microbes. And this obviously provides a more holistic view. So they're looking at the hologenome, which encompasses the entire genome of the animal plus all the symbiotic microbes. The study specifically wants to look at the coevolution of the diamondback moth's gut microbiota along with the moth itself, because there's a theory that they co-evolved essentially in order to help the moth adapt to its environment and also arise as such a great pest and herbivore. But it also wants to look at what specific bacteria, and although it's going to get a holistic profile, it wants to look at the specific bacteria in the moth's microbiome and which of those play the greatest role in those herbivorous mechanisms. And of course, after that, it's going to pave the way for new research to attack these bacteria and their mechanisms. And of course, the specific bacteria are going to come with enzymes and certain proteins that participate in these mechanisms, and these are going to be the main targets for attack. So onto the methodology used in this study. So first, the insects had to actually be reared. So a certain strain of the Plutella zellostella, which from now on is going to be just referred to as the diamondback moth, were collected actually in July 2004 from a vegetable field in Fuzhou, China. The larvae were fed radish seedlings at 25 degrees Celsius in 70 to 80% relative humidity, and a photoperiod that consisted of 16 hours of light and 8 hours of dark. So 2/3 of their day in light, 1/3 in dark. And most importantly, the insects were given no exposure to insecticides. The pupae, after the larvae matured into pupae, they were collected and then they were put into a 500 milliliter plastic bottle. And then once they grew adults, they began being fed a 10% honey solution, which encouraged them to mate and finally lay eggs. And finally, the neonates, which hatched from the eggs, which at this point were the pure generation that was born during the study, they were passed through holes in the bottle and they dropped onto the radish plants. So 200 individuals each for 3 different developmental stages, one being the third instar larva stage, the other being a pupae, and the other being adults. So 200 from each of these 3 groups were randomly picked and sampled, no matter the sex of the insect. They were sterilized, dissected, the gut contents homogenized with a milliliter of sterile DI water, and they were frozen at negative 80 degrees Celsius for DNA extraction. And so the QI-AMP DNA stool mini kit was used to actually extract the total DNA from all the samples, and then it was run on 1% agarose gels and recovered. So finally, the most important part of the methodology is actually the construction of the DNA library and the actual sequencing, and this obviously correlates to our class, the microbiome. So a paired end library was created and the insert size of each of the DNA sequences was 350 base pairs for each life stage sample. High throughput sequencing was done using Illumina HiSeq 2000 to obtain the metagenome data. So each of the paired end reads had a length of 100 base pairs. Some of them had an N, meaning that any of the 4 bases could have been there because it was inconclusive. Usually it's caused by weak signal during sequencing. It can also happen if the read length is too long for the technique being used. So these were discarded if they had an N, if they had 3 or more N's, meaning that it was unclear what bases they were, or if there was adapter contamination. So the reeds that were contaminated with the host genetic material were also removed.